Okay, hello. So this is the continuation video of apoptosis. In the last video, we studied the basic of apoptosis. Now, we'll be dealing apoptosis in more detail. So the images in previous video and this video are taken from Wikipedia. You can even look out for references. Now, uh, when dealing with apoptosis, so apoptosis we'll be dealing in little more detail. Apoptosis is a Apoptosis can follow two pathways. One is the extrinsic pathway, and another one is the intrinsic pathway. In extrinsic pathway, the external signals will uh, allow apoptosis to occur, and in intrinsic pathway, there are no external signals, rather than there are internal signals. So, first we'll study the extrinsic pathway. Extrinsic. The diagram which you see here represents the extrinsic pathway. So this is the cell membrane. Our cell membrane have various receptors, okay? So one receptor is tumor necrosis factor. So whenever the cell is damaged or unhealthy, uh, a cytokine called as tumor necrosis factor, okay, goes and binds to the tumor necrosis factor receptor, and this will cause the changes in the cytosolic death domain, and this changes in the cytosolic death domain will allow the interaction of two uh, proteins called as TRAD and FAD. Actually, this TRAD is a component of this cytosolic death domain only. So, interaction of these two proteins will recruit procaspases. Procaspases is the caspases in its inactive form. This is cleaved into uh, four into a tetramer, into an active tetramer. Now, active tetramer is called caspase 8. Now, these caspase 8 will in turn activate executioner caspases, and that executioner caspases will carry out apoptosis. And the function of caspases has already been discussed in previous videos. So this is the extrinsic pathway of apoptosis. We need to remember that caspase 8 is involved in apoptosis of extrinsic pathway. And one more point I left out in previous video is actually what is the component of caspase 8? Caspase 8 is made up of actually it is a enzyme, means it will be made up of proteins, but it is also called as cysteine aspartate proteases because it has cysteine aspartate in its active site. Remember this point, this may be important. Okay, so this was extrinsic pathway. Now next we'll study the intrinsic pathway. Intrinsic pathway is actually very important. So before studying intrinsic pathway, we need to know certain proteins that are present in healthy cells and how are they interacted with other proteins. So first is there are certain pro-apoptotic factors or proteins, and another class of proteins is called as anti-apoptotic proteins. Pro-apoptotic proteins are the proteins that actually promotes apoptosis, and anti-apoptotic proteins are the proteins which inhibit apoptosis. So example of the pro-apoptotic proteins are PAX, protein, back protein, and anti-apoptotic proteins example are BCL proteins. One example is BCL2 protein. So we need to remember that pro-apoptotic proteins promote apoptosis, anti-apoptotic protein inhibit apoptosis. Now in a healthy cell, there are both pro-apoptotic and anti-apoptotic protein, but yet uh, this pro-apoptotic protein will not cause apoptosis because the cell is healthy. Why? Because this anti-apoptotic protein like BCL2 protein will go and form heterodimers with anti-apoptotic. This BCL2 is an anti-apoptotic protein and this PAX is a pro-apoptotic protein. So this BCL2 will form a heterodimer with PAX protein and thereby inhibiting apoptosis. Okay, uh, and what happens in unhealthy cell or stress cell? In unhealthy cell, there is overexpression of PAX protein. So PAX protein forms homodimers. This is one PAX protein and this is another one PAX protein. They form homodimers and this will promote apoptosis. Now what happens in case of one interesting concept is what happens in case of cancerous cell, because as we all studied from high school, cancer cell are called immortal cell, right? Why? Because they do not die. So in cancer cell there is high expression of BCL2 proteins because they are anti-apoptotic proteins. And basically cancer is a protein which does not require apoptosis because they are immortal. So BCL2 protein are overexpressed and two BCL2 proteins will form homodimers and inhibit apoptosis. Okay, this is the thing that we need to remember. Proteins, pro-apoptotic proteins, they promote apoptosis. Anti-apoptotic protein, they inhibit apoptosis. Examples of pro-apoptotic protein are PAX and BAC protein, and anti-apoptotic are BCL2 protein. In a healthy cell, there is an interaction between these two pro-apoptotic and anti-apoptotic protein, and they form heterodimers and they prevent apoptosis since that is a healthy cell. And in unhealthy cell or stress cells, there is overexpression of pro-apoptotic protein because the cell is unhealthy and it has to undergo apoptosis. So both of the PAX proteins will form homodimers and promote apoptosis. Cancer cell, in other hand, is seen to have overexpression of anti-apoptotic protein because they are immortal, and this overexpression of BCL2 protein inhibits apoptosis. So in the next video, I'll be dealing the actual pathway of this intrinsic pathway of apoptosis. Okay, thank you for watching and hope you are understanding my concepts, and please do like, comment, subscribe to my channel. Thank you.